From Studio A at the USC Annenberg School. My name is Krish Wadhwani, and this is the Paradigm Podcast by Shift SC. Today we have Travis Williams, who is an OChem professor at USC, and he's going to be talking about how he recycles plastic using a strain of fungi. So, Travis, tell me about how you started in the field of organometallic chemistry.
Sure. So when I'm not doing this, I teach the organic chemistry class, which it's the one everybody's afraid of. The pre-meds have to take it, et cetera, et cetera. And one of the things you learn quickly when you sit down to take that class is that it's really not like any other STEM class that you've encountered, because it has this big component of visual manipulation. You see structures, and we start talking about shapes, and at the same time that we're having chemical reactions happen, and there's kinetics and thermodynamics, all the quantitative things that we browbeat you with in freshman chemistry, you get to talk about shapes and structures and functions. And when I first took organic, this to me was like waving the red flag in front of the bull, because here's a STEM class, really is a STEM class that's more drawing intensive than equation intensive. And I latched onto this quite readily. And you run into a limitation, though, as you start probing the structural novelty that you can achieve with organic molecules, which is that there's 4 bonds to carbon, and there's not a lot of ways to change that, right? And the bonds have a certain strength. You know, it's about 90, give or take a few kcals in the first row of the table. And so if you wanted to do fragile, transient, or dynamic things, you'd need something that's not a first row element, right? The transition metals, we're talking anywhere from 1 to 9 bonds with bond strengths that go anywhere from a little to a lot. And so when you bring the transition metals to bear on organic reactivity, you open up a whole lot of new possibilities that the community didn't begin to discover until, I'd say, about the 1960s. And it was in my lifetime that the transition metals started to propagate through the organic community. So when I was your age, this was still relatively new.
Yeah, that sounds beautiful, especially all the 3D designs and the reactions and all. And honestly, I might change my major because I like the way you put it. So I think today we want to focus on, this method you've devised to degrade plastic, because there's like millions of tons of plastic everywhere, and I don't think I need to go on about how plastic affects our lives, but just about like, what have you devised to degrade plastic?
So what are we doing? Okay, so let me present you this problem. We made these molecules because they are robust, right? We made these molecules because you can't break them. And the American Chemistry Council, not to be confused with the American Chemical Society, the American Chemistry Council, which is an industry lobbying group, back in the late 60s, early 70s, when we were trying to promulgate thermoplastics into the economy, they invented the chasing arrows diagram. You've seen the little recycling symbol. They invented that, and they sold this bill to the American people, which is, well, you can melt process these, they're going to be recyclable. They didn't tell you sort of the facts of the matter as to the limitations of that kind of approach, or that trying to recollect these and get them clean was never going to be practically achievable. Because that we designed them to be robust, we designed them to be abiotic. And so there is no biodegradation pathway for these materials, which is great if you're trying to make like medical devices, right? They'll last forever. Body can't break it down. Not so great once the American people bought this bill of goods. We started making these incredibly inexpensive, robust, useful materials, and then they start accumulating in the environment, right? Now, I don't know that I have a real problem with sending plastics to landfill. I mean, fundamentally, it came out of the earth as petroleum. We're putting it back in, right? But some fraction, it depends on who you ask, between like less than 0.1% up to a couple of percent of our municipal garbage leaks into the environment. And you've seen it happen, right? Garbage trucks rolling down the street, picks up the can, and plastic bag blow. That's what I mean by leaks. And then this ends up in waterways, and this is the problem. there's no biodegradation. I mean, we engineered it to be that way. The chemical degradation, I mean, we could degrade anything, right? I mean, we got the hammer, but if you take a plastic, and let's talk about polyethylene because that's sort of the big boy, right? Structure of polyethylene is a chain of carbon atoms, CH2, 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 polymer chemists into chains, all right? And if you try to blast that, where is it going to cleave? It doesn't know either.
Wait,
right?
What do you mean by cleave?
Okay, so if you got a polymer chain,
Yeah.
right, and every element in the chain is the same, where is chemistry going to cleave it? It doesn't know either. It's here, right?
Yeah.
This isn't like genetic technology where there's a DNA sequence, you can read a piece of the sequence and right there, that's where I'm supposed to cut it. It's just going to blow it to pieces. And so what you're going to make with any cleavage technology that comes out of a chemistry strategy is you're going to make a diverse host of other waste materials, right? You're not making a product, you're making another waste material.
Yeah.
The idea that Clay and I brought to bear on this is the biology is never going to be reactive enough. This is an abiotic material. So and bless them their effort to go send microbes to go remediate oil spills. It's just never going to happen on manufacturing time scale, right? It's going to happen on environmental time scale. Can we use chemistry to blast the plastic into something that looks biological so that we could confuse the microbe to think that it's food? Then we could do fermentation-based manufacturing. A whole lot of things you buy are fermentation manufactured. You aware of that? Beer, right?
Yeah.
Yeah, that's a yeast. A lot of your antibiotics are manufactured. There's some big tank that looks like a brewery, except it's making erythromycin, right? Can we somehow blast the plastic into a material that a fungus or a yeast or a bacterium would recognize as food, build one of those big brewery-looking apparatuses, and have something come out the back. It's probably not food because it came from like trash, right?
Yeah.
Have something come out the back that's really valuable.
So you used fungi, the strain of Aspergillus nidulans, to basically decompose plastic. So, you said that plastics are structurally sound and are indestructible and have these long chains. So is there something about fungi that is similar to plastics or maybe completely opposing poles to it that makes it such a good candidate to degrade plastic, and not only that, but come up with useful, viable byproducts?
Well, we're still learning. Aspergillus nidulans is a single-cell filamentous fungus that grows everywhere. It's very easy to proliferate. You've got some plants growing here in the studio. That's nice. These are reasonably easy to proliferate. You can water it, it'll grow. You've tried gardening. Some plants, doesn't matter what you do, they're going to die, right?
Yeah.
Not nidulans. Anybody can grow nidulans. And over the years, even before we had CRISPR and the modern genetic tools that we have now, early restriction endonuclease technology was sufficient for people such as my partners, Burl Oakley at Kansas, Clay Wang over in our School of Pharmacy, worked out chemical methods to use early genetic tools that you can drop genes and gene sequences into Aspergillus in predictable ways. And they're not the only people doing that. I mean, the way we make lovastatin, that's a cholesterol-lowering drug, is similar, right? We're fermenting molasses using Aspergillus terreus, and it expresses lovastatin, right? And that's a natural process, but they engineered it to be more productive. So Burl and Clay and their competitors over 20 years have developed really easy to use, really effective ways to drop naturally occurring gene clusters from other organisms into nidulans, basically turning the everywhere fungus into one of these fermentation factories. And you can pick a gene cluster. So like how did we discover penicillin, right? It came from mold. We discover a lot of natural products and medicinals this way. Used to be we'd have chemists go figure out, well, what are the molecules that do in the biology? We do it differently now. We look at what are the genes in the organism that's making this thing that does the biology. We know enough genetics to know, okay, these are the genes that have the magic. We have the tools that we can excise those gene clusters, or we can sequence those gene clusters, replicate them synthetically, and then take that piece of DNA, use the tools that Burl and Clay and the Nigellans community has developed, drop it into this fungus, proliferate the fungus, and it'll just spit out the product that was encoded in that gene sequence.
Okay, just sidetracking away from plastic degradation for a second and going into gene editing specifically, and maybe certain mutations that might come up, say, with your genetically modified Aspergillus nidulans that may mate with, say, a regular strain of fungi. Is there something called like hybrid vigor that might like make this monster? Or as we
Maybe.
know, like The Last of Us.
So humanity had to confront this in the 1970s, right? The humanity had to confront this in the 1970s when, for the first time, we had tools where we could excise a gene cluster here, there, something. At some point, science has to be regulated, law has to intervene, right? And the first time in the history of the U.S. that happened was the city of Cambridge, Massachusetts, in the late 70s, had an ordinance where they outlawed any kind of recombinant DNA technology. This is noteworthy because Harvard and MIT are located in the city of Cambridge. And so there was this very public, very colorful debate in Cambridge City Council as to whether this should be done and how it could be done safely. I encourage all of your viewers, go watch the tapes of the Cambridge DNA debates. Knowing now, 50 years later, what we know, it's amazing. Okay, cool. So what follows from that is, use and leverage of NIH funding, the government requires institutions that manipulate genetics or biohazardous materials to self-certify the safety of what they're doing and classify according to well-designed criteria. Is it possible that this could be the end of us? And then you kind of grade the technology biosafety level 1, which is where we are. All of our materials are BSL level 1 materials, which are on the same danger level as beer making.
Right.
Right. And then BSL 2 could potentially be infectious. BSL 3, could potentially kill us all. BSL, I don't know how high it goes, but we don't go beyond 3 in the USC campuses.
So looking at the state of bioethics right now, we have like biohackers like Hosea Zeno that end up taking power into their own hands and basically editing their own gene. Like, this dude has changed a gene for muscle growth at a conference where he said that, this is my genome and I'm in power of myself, so I can change my own genes. And there's this whole community of biohackers that feel that true freedom is editing their genes and maybe giving themselves superpowers like glow-in-the-dark skin or like hypersensitive
That'd
hearing.
be cool, right?
It would be, but like I'm trying to think of like what's right and like where's this all going?
Yeah, so there's always going to be bad actors, and there's always going to be a need for government and the public to understand and then legislate the promulgation of technology.
Right.
And that's going to be true forever, in all areas.
So, are you for or against people taking the liberty to edit their own genes and give themselves superpowers? Because UC Berkeley is already doing it, right, with the DARPA program where it's militarized and it's public funding where they're trying to make super soldiers, and so it's already happening. But could this also just be another stream into like enhancements and people just changing all sorts of things about themselves?
I would say that it strikes me as a bad idea to be manipulating chemical or biotechnology outside of an environment that is very well peer supervised. Now, the University of California is an environment that's very well peer supervised. I also think it is generally a bad idea that we should be doing those kinds of things in secret. So I think humanity and technology are best served when this kind of work is done in a knowledgeable, peer-supervised environment, regulated by the systems we have in place and observable by the public.
Okay.
And I think if we do that, we will make good ethical decisions going forward.
I agree with that, and like yeah, I also think it is a bad idea, but I really think that's where the profit mechanism comes into play, and if like if people make it lucrative enough, then people are obviously going to do it, but there needs to be certain legislation to stop that and interfere with the profit mechanism. Anyways, the last time I spoke to you, you said that with your fungal engineering idea with Aspergillus nidulans, to genetically engineer them to decompose plastic, you don't really use CRISPR or talons, but you've actually devised your own technique, which is old school,
Yeah.
but works nonetheless.
Yeah, and let's credit the folks who did it. I'm working with Burl Oakley and Clay Wang. They study this, and they developed a lot of those tools in their own laboratories. I break molecules. They selectively modify the genome, and they could tell you more about how those tools work. But they predated the modern CRISPR methods.
Okay.
The CRISPR methods work, and I know that Clay and my colleague Peter Chin have used CRISPR-Cas methods to make nidulans. It works quite well, as I've seen. But it's what I understand from the people who do those experiments is that they are difficult experiments to do. It's like a Formula One race car, incredibly powerful tool, right? But not easy to use, not easy to maintain. As I understand the existing technology of nidulans, it's just really robust, and it works really well.
So you have a research team of students here at USC
I do.
that help you, sort of engineer this idea.
Yep.
So could you tell us about some of the work that you guys have done, whether it's at USC or in Catalina?
Yeah, so the team is very diverse, right? We've got chemistry students, graduate students, and undergraduates who are working on the problems of how do you safely, scalably, selectively chop up plastic, and what is the right material for us to make to create things that the fungus wants to eat. How do you adapt these methods to the different plastics that are out in the world, right? We're struggling right now with polypropylene. We can get it to work, but it's not easy, right? Not like polyethylene, which, I mean, it's like falling in love. The fungus will eat that for nothing, right? How do you deal with it when your material came out of the ocean and is populated with salts, tar, sand, barnacles? It takes a chemist to look at that and figure out how can we do this? How can we clean it up post cleavage? How can we get and the students who are doing that are anywhere from some undergrads. I've had postdocs work on this. The lead, as in any kind of project we do in USC, the lead is going to be a graduate student. Yu Hao Chen, heck of a guy, has developed a lot of this. I have a team of undergraduates, always recruiting for this detail. I have a team of undergraduates who live out at Catalina over the summer. They're mostly federally funded with education money, with the task of teaching the public about chemistry and sustainability, and we embed them in a Boy Scout camp out there. And they teach merit badges, and while they're out there, they source feedstocks. And they bring us ocean-sourced plastic, used motor oil, fiberglass, things that we wanted real environmental source samples to demonstrate. So they take care of that for us while they're out there.
That's amazing. That's
if you're interested, let me know. I'm always hiring for that detail. And then we have at the School of Pharmacy in Clay's lab graduate students, postdocs, collaborators, visiting scientists.
So going back to how we started this podcast, where you described plastic as this like indestructible material.
Yeah.
So when designing fungi, do you think in terms of like this might be a biophysics question, but do you think fungi needs to be like also indestructible? Like what can we change about fungi to make plastic its food?
Got it.
In like
so
a little
so
child's language.
this is a research strategy question. Okay. Do you want to change the organism to metabolize the feedstock, or do you want to change the feedstock so that the organism will metabolize it?
Well, here we can't change the feedstock.
Yeah, I can. I'm a chemist. I can change stuff.
Okay.
Right? Okay, synthetic chemists have the ability to convert one substance into another. This is what we do. And what we're doing with polyethylene is we're chopping it up to make it look like a fatty acid, make it look like food. Okay? I would rather use that strategy for two reasons. One, if you try to decode and manipulate the biological system is always more complicated than you expect, right? And our competitors for a long time have been trying to do that and haven't nailed it yet. Okay. The other one is I get just a little bit nervous about some of these bioethical issues that you raise about if we make an organism that can metabolize anything, should we be just a little bit worried about that? Right. So I would rather whip some chemistry on the trash where I know the chemistry is going to be contained into some sort of a refinery and is not likely to get out, and I know what can make it explode, and I know what it's going to cost and how scalable, et cetera. I'd rather engineer that than engineer the organism. I'd rather have an engineered reaction, chemical reaction, than an engineered fungus.
In one of your research papers that was published this year, actually, I think you spoke about how the byproducts of like when the fungi feed on plastic can also be proteins that come up from the engineering process.
Or whole organisms. Oh, yeah.
Oh, whole organ. Yeah.
Yeah.
Okay. Wow, I'm still wrapping my head around that.
Yeah.
So, like, in terms of like coming up with proteins, are you saying that the byproducts of plastic could be consumable proteins, or?
Oh, yeah.
No way. Sorry, I
Do you eat fungi in your life?
Yes, I love
Yeah.
mushrooms, yeah.
Okay. So what if we grew those on post-consumer plastic? No, I'm not real keen on that, right? Because
Why not?
Because we have other good ways to produce food. But let me highlight some things where the fungal products enter your life. What do you know about laundry detergent? How does laundry detergent work?
Well, I put a
You put a pot in the machine, it comes out clean.
Yeah.
If you go shopping for laundry detergent, next time you go, take a look at what they cost, okay? There's some of them, and I'll call it like Target brand, that are pretty inexpensive. And there's some others, and
That's
I'll call
the one that I get, the inexpensive one.
Yeah, I go both ways.
Okay.
There's that one that's inexpensive, and then there's this like Tide or something that's considerably more expensive. There are a number of really cool biotechnologies that are engineered into laundry detergent. The cheap one is soap, right? And we all kind of know how that works. It's an aqueous organic compatibilizer that takes the grease and solubilizes it and goes down the drain. No problem, okay? But the biotechnology is, wait a second. What if we unleash an enzyme on the soap, cleave the glycerol off of the triglyceride, then it's water soluble without ever having to unleash the soap on it. Soap is a negative environmental impact. They don't biodegrade. We don't particularly want them in the ocean. So what if we displace the soap from the laundry detergent with nature's approach, which is enzymatic cleavage of the fats, and solubilize them in that way? Now what we're unleashing on the environment is amino acids that came from that protein, fatty acids that came from the dirt on your clothes, and glycerol. Well, those things are going to be in the environment anyway. The fish are going to eat them. Great, okay? So manufacturing that enzyme is very difficult relative to soap. I mean, soap is a petrochemical. We know how to make it. But trying to make something as complicated as an enzyme involves biotechnology. You got to encode that genetically in an organism and make a fermentation reactor, isolate it, clean it up, and then sell it. And the enzyme has to be stable to all of those processes, in addition to shipping and distribution of the product. Okay, so let's make that out of burnt plastic. Because you're not going to eat it. It's an environmentally clutch technology. Because it's displacing a persistent pollutant from the ocean.
Yeah.
This is where I want to go, right? there are biomaterials. So there's an organism, Bassiana, B. bassiana, which is an infectious disease. It's a fungus. It's an infectious disease that infects aphids. Aphids destroy agriculture. They destroy crops. We don't like that because we want to make money off agriculture and we want to eat, right? So what are your choices? Your choices are, so here come the aphids, you know, thirsting for corn or whatever. Your choices are you spray a chemical insecticide on the farm, which will end up in the waterway and will have secondary impacts on the waterway, none of which are likely to be good, right? Or can you proliferate a culture of Bassiana in the farm where that organism, as soon as the aphids come, are going to infect the aphids, eat their brains, and then you have dead aphid, right? I can handle dead aphid in the water supply a lot better than I can handle carbon-chlorine bonds, right? So let's make Bassiana out of burnt plastic. It will intersect the food supply.
Speaking about food, you know, one of the other byproducts of this reaction with nidulants and plastic is also benzoic acid, which is just this crucial precursor to so many different products.
It is the longevity of the Twinkie. Read it, potassium benzoate. That's how the Twinkie lasted through the Ice Age.
Okay, I like Twinkies.
Approved for human consumption, but I don't recommend it.
Right, so
watch out for the Twinkie.
Coming back to benzoic acid, you know, what can we use benzoic acid for, which is like this byproduct of the reaction?
Yeah, I don't know. So if you put on your entrepreneurial hat, the answer to that question is go ask the marketplace what it sells for and what the market clearing quantity is. It's cheap and there's not a lot of it sold. It is going into the food supply in the form of potassium benzoate. It's a preservative. It's an antioxidant. We don't like that as a consumer product because it's not expensive enough. So we got a lot of competitors in the chemical space that are burning down polystyrene to make benzoic acid. Hats off, some of these are great methods. Some of them are very selective. I claim ours is just a little bit better, but nobody cares. What we did that mattered was we figured out a way to take benzoic acid and to turn it into something that really is expensive, that we really do need, which is resistant cell line antibiotics, biocontrol agents to displace pesticides from the farm, and biomaterials.
And one of these, you know, antifungal agents is for aflatoxin.
Yep.
Could you also just go over, you know, how aflatoxin affects us, and if this works, we could turn plastic into different like useful things that end up
bingo.
Curing
and
us.
This would be really cool if we could explain this to your viewers. Bassiana. Okay, this is the.
What is Bassiana?
This is a fungus this is the infectious disease that kills the aphids.
Okay.
Okay. So conceptually, what's happening there? We took a fungus that we know to be an infectious disease. We turned it loose on the farm, and we killed a bunch of aphids without hurting the corn or the cotton or the strawberries or whatever, spinach. Spinach is a tough one here in California. How do we feel about that? Well, you know, I'm not so keen on unleashing infectious diseases, but I mean, this was natural. It's been around forever. And I feel a lot better about this than I feel about putting chlorocarbon pesticides into the water supply. Okay, so this is a concept in agriculture that they call a biocontrol agent. Okay, biocontrol agent is what happens when we take a natural organism and we use it to suppress other natural organisms to the benefit of our target product. Vegetables. Okay. AF36, which we mentioned here in this Jack's paper, there's a fungus, Aspergillus flavus, that makes a side product, a family of side products called aflatoxins. Aflatoxins are nanomolar hepatotoxins, which means very dangerous for the liver. People die of aflatoxin poisoning at very low levels. Flavus, much like Bassiana, So Bassiana is an infectious disease, right? It doesn't infect us, it infects the aphid. Flavus is an infectious disease that infects corn, peanuts, tree nuts, and cotton. If you have a flavus infection in your cornfield and somebody eats the corn, they could die from aflatoxin poisoning. And people do, but not in the United States, because the USDA is good enough at recognizing fungal infection in the food supply that they'll just burn down the cornfield. And that's good, right? Because then nobody dies. But we're burning on the order of $1 billion of US corn every year to keep aflatoxin out of the food supply. And we should. Okay, cool. So about 5 years ago was characterized in Arizona a strain of flavus, Aspergillus flavus 36, we call it AF36, which has a polymorphism in the second gene of the aflatoxin cluster. Genetically, that means there ain't nothing going to happen that it could ever make the toxin. This is non-toxic.
Right. Yeah.
Great. Because it's non-toxic, it proliferates better than the other kinds of toxic flavus. The FDA and the EPA have approved and encouraged the use of AF36 spores as a biocontrol agent. You can inoculate your seed corn with AF36 spores, they'll proliferate, and then toxic flavus cannot infect because it's been out proliferated by the non-toxic flavus. Cool. So we've introduced a manufacturing process to make AF36 from styrofoam.
Plastics.
Yeah.
And just to zoom out a little bit here, your researchers said that you get approximately somewhere between 15 to 20% molar yield from the plastics, from your research that you did with the plastics that accumulated on Catalina Island.
Yeah.
So my question is this scalable? Like, could we grow this? Could we grow fungi on all different types of plastic and basically be the end of plastics and great ocean garbage patches?
Yeah.
What's
So think about it like this.
Yeah.
So you go down to the port and there's all these petroleum refineries, right? There's going to be an operation that looks like that, and it's going to be running these kind of chemical reactions that chop up plastics. There's going to be huge safety concerns and engineering and planning, and just like you would in any petroleum refinery, where we're taking this petroleum product and we're backing it out into another petroleum product, which is what we do. And you can build more of those refineries, and you can build bigger refineries, and you do not want to put it in downtown LA because it's chemical manufacturing, right? And then there's going to, from that plant, then we're going to load up this, I will call it toxic sludge, into some sort of truck, right? And we're going to drive it to another refinery that looks an awful lot like a brewery. And we're going to dump this truck into these giant tanks in this brewery that are full of...
Nidulans.
Exactly. And there's going to be a different nidulans in every tank, right? And then we're going to heat it up to 37, same temperature as you and me, right? We'll stir it for like 2 days, open the tap, and out's going to come medicinal products, enzymes, biocontrol agents, things that we can sell, right? Things that are very expensive that humanity needs, and we're kind of emphasizing, let's very first thing, let's do things that protect the food supply and protect the water supply.
So what does a roadmap look like for something like this to really be feasible and become a reality? Is it, I mean, obviously there's funding involved. So what type of funding?
So what we're doing now with this is we're looking around. There are big companies doing large scale fermentation manufacturing. There are and they're very good at it, and there are large companies doing oxidative catalysis based manufacturing, and we're trying to recruit some of these partners that we can, for what I'll call a relatively low price, sell the USCIP to them, license it to them, sell them some consulting hours, we'll help them do it, and can we get another company to take this, run with it, scale it, and deploy it? And I'm guessing that's not going to happen because businesses tend to be risk averse, and to anything, even HR changes, they tend to be very risk averse and not so entrepreneurial or agile. So I think what's going to happen is we're going to have to go out in the marketplace, just like we did with glycerol to lactate, just like we did with recycled aircraft, and we're going to have to find the product, find the customer, and find the price point where we're going to sell something. Let's say it's AF-36, and we're going to figure out who's going to buy it, how much can they buy, then we're going to have to recruit venture capital, then we're going to have to build a refinery, probably about the size of the studio. So, I mean, that's a reasonable couple $1 million, right?
Yeah,
Yeah,
and that's a small amount compared to how much America has designated towards climate tech. Like it has a budget of over 300 trillion,
that's right.
and if like a small percent, like
Yeah.
under 1% of that is
Yeah.
directed towards this,
Yeah.
that could be the end of plastics as we know it.
Well, I mean, so first we got to make the refinery the size of this room and make it work. But then at that point, I think you've de-risked it to the point that the Dows and DuPonts and Cargills and P&Gs of the world take notice and buy out that startup. And then you watch your baby fly, right?
Yeah, it's VC funding, I think, like you said.
Yeah.
That's what it really.
But you
comes
got
down
to,
to.
yeah, you got to make that step. And like I implied, I've made that step with a couple other technologies out of our lab.
Tell us about that, please.
Okay, so what's killing us in both of them is the people. You got to have people who have the stomach to take on that risk straight out of graduate school, and you got to find the right CEO, and then you got to de-risk through the middle technology readiness levels. You got to scale reactors. You got to constantly recruit venture capital, and you got to constantly negotiate these sales contracts where a risk-averse company is going to change suppliers for a known material, right? Or a risk-averse company is going to buy several kilograms of something from some third-party new player where they're used to buying hundreds of kilos a week, right? Why would they mess with that? And so trying to break into large-scale industry as a startup just takes persistent pressure. And that's beyond the scope of academics to go out and do that. So we got to recruit really robust students and young people who, the hair on fire and crazy in their hearts, who want to take technologies, scale them, and make those arrangements and cause it to happen.
Yeah, and there's also just a perspective shift that I think needs to happen, which is like this symbiotic relationship with nature.
Yep.
Which, I mean, there's a few companies out there that are already doing this with Neri Oxman's company, who I think she focuses on just this emphasis on natural design and coming up with different materials and biomaterials, like building houses of cellulose and all these like organic materials. So I think it's really just like forming a relationship with companies like that, and which companies that have an emphasis on changing the perspective, especially with an idea like this. So I think in terms of like concluding remarks, like I'm thinking about, are there any calls to caution with fungal engineering, and is there any like downsides to this? Because I'm not hearing any downsides. I'm hearing like different byproducts that have all these different uses, from agricultural to pharmaceutical to consumptive properties.
So in point of fact, we've been manufacturing stuff by fungal fermentation for many years. And this looks pretty good to me, right? As manufacturing strategies go. I think the caution that you're right to raise is that as we are genetically engineering new organisms, we got to do that in really well supervised environments where smart people are thinking out what are the potential consequences here.
Yeah, and I think that's really with anything. I think if we have our main mission and our main goals that are clearly highlighted, clearly stated, then we just need to stick to those ideals and really get rid of this plastic problem.
Yeah.
Which I think, the reason that I invited you here is because I think that's a viable solution.
Yeah.
And I just generally resonate with the work that you're doing.
Yeah, thank you. Appreciate it. The line I used with ABC was we committed a certain amount of our petroleum resources, which are valuable, into making plastics. And there's energy and there's carbon atoms in those resources that we're sending to landfill, and frankly, I want it back.
Yeah.
Right?
Yeah.
I want it back.
What you're saying is there's money in the landfills, basically.
There's money in this material, right?
Yeah.
And it presents in the form of energy and in the form of food. Not for me, but for somebody, for Anigilans.
Yeah, definitely. Well, also, I'm also just interested in the metabolic pathways of fungi and just like uh, how they end up like degrading. Like, what do nidulans usually eat if it's not plastic?
So we're manufacturing stuff by fermentation on large scale, and what the first thing that we would choose to feed that is molasses. So we're producing sugar, right? A lot, some in the United States, a lot in Central America. We produce sugar, and there's a sugar cane. You grind it up, and you do an extraction, and you extract sugar out of that. And it's an aqueous solution. You heat it to drive the water off, and when you do, you it's like cooking something. You kind of brown it. And then they do this recrystallization where they crystallize sugar, and that's the white powder that you buy in the grocery store, and I call that food. Okay, great. And then they recrystallize it a second time, and then that's got some of this brown toasted stuff in it. We'll call that brown sugar, all right? And that's, again, I'll call that a type of food. You should eat that. And then after that, you can't crystallize anymore, and there's this kind of brownish, blackish, tar-like liquid. They call that molasses, right? That's also a type of food, but it's used a lot less, right? They take molasses and they dump it into the fermentation reactor. So it's like the dregs of sugar that we weren't able to sell otherwise. And there are a lot of products like that. There are a lot of products like that out there in the economy that we were producing a food from a plant, and then there's all this stuff that's left over that's full of CH bonds that contains energy and et cetera. Molasses is an example, and we manufacture it into fermentation products. Another great example is soybean oil.
And all of this is essentially just natural hydrocarbons?
Natural products of this, that, and the other kind. You know what soybean oil is. You know, Smart and Final, buy a can of cooking or a bottle of cooking oil. It's dirt cheap. Why? Because it's surplus. This is soybean oil. We know how to turn that into diesel fuel. And we should, right? Yeah. So we should definitely be growing soybeans. We should definitely be harvesting that bushel. In the bushel of soybeans, there's 80% of what comes out of that bushel is going to be soy meal, and that's going to be tofu and edamame and all of the kinds of stuff we love to eat, and 20% is going to be soybean oil. Some of that we're going to sell for like deep fryer and whatnot. Most of it we can't sell. So what do? They take a little methanol, chop off the glycerol piece that holds it together, make these fatty acid methyl esters, pump it into the gas. That's biodiesel.
So I'm just going through your research, and I see that there's a line here that says that the differences in recoveries can be explained by the presence of additives in plastics such as free radical scavengers and composite polymers. So my question is, do we need to change the way that we make plastic? It might be too late for that because there's so much of it.
I'd rather we didn't, right? Yeah. So we engineered the plastic to do something that was important, which is why we engineered it in the first place. I would rather, now that we're going to go solve this problem, I would rather engineer a solution that accommodates the materials that occur in nature. Let's talk about polyethylene. Polyethylene is a brittle, difficult to process solid. And so we do things like we put dioctyl phthalate in it, we put tougheners in it, we put other polymeric materials in it in order to make it so that we can melt process it into toys and tools and devices and the chair you're sitting on, right? I think so. Then when we take these post-consumer products, we chop them up. We chop up the polyethylene. We chop up all the other crap that's in there too, right? Now we don't know what it is, but we don't care, right? Because we chop it up into these carboxylic acid fragments that more often than not the fungus recognizes as food. It just we don't know what it is, so we don't know how to count it. So the mass recovery appears less than it actually is because we can't say this is this. It's just some mass, and much like the molasses, who knows what's in there? It's like half toasted sugar, right? It's a bunch of stuff, but the fungus loves it, and off it goes making enzymes and medicines and all sorts of other stuff.
Yeah, I think I read somewhere that I think in about 100,000 years, we may not be there, but there's definitely going to be fungus that lives and grows. So how is it that, fungi are just like so resilient and just be able to withstand any sort of conditions? Were they like, I'm not too sure about my biological history, but were they the first organisms on Earth?
I don't think so. I kind of think that was Monera, but I'm not a Monerans, but I'm not a biologist. I'm going to appeal to my automotive analogy. There's your Honda Civic and your Formula One race car. Okay, you are the evolutionary race car, but you have a lot of environmental demands. You have to be stored at a particular temperature, fed a particular food supply. You have to have a certain humidity level, amount of water, a certain amount of sunlight, but not too much. The fungus is more like the Honda. You just drive and it goes.
It's resilient.
It's
got good
requirements. All right, got good mileage. Its requirements to thrive are a lot less.
Yeah, I don't want to take too much of your time, so I think that is it. But
Rock and roll.
thank you so much, Travis.
You bet.
This was
Thanks
great.
for the opportunity.
Thank
Happy
you for
to
your
chat.
time. Yeah. From Studio A at the USC Annenberg School. Thank you for listening to the Paradigm Podcast by Shift SC. If you liked it, please subscribe. You can catch us on YouTube and Spotify.